Good morning, everyone. My name is Xuan Bao, and I'm a postdoc from Dr. Ray Chen's lab. It is my great pleasure to introduce today's speaker, Dr. Dmitry Ivanov, professor of ophthalmology, microbiology, and immunology at the University of Miami Miller School of Medicine. Dr. Ivanov received his master's degree in biophysics from the Moscow Engineering Physics Institute and his PhD in molecular genetics and molecular biology from the Vavilov Institute of General Genetics of the Russian Academy of Sciences. Before joining Bascom Palmer Eye Institute, he contributed to major international research efforts, including the discovery of genes associated with B-cell chronic lymphocytic leukemia and the cloning and characterization of the pannexin family of gap junction proteins, a field that has since generated over 1,000 publications. At Bascom Palmer, Dr. Ivanov has built a highly impactful research program focused on the molecular and epigenetic mechanisms that govern neural development and degeneration in the retina. His laboratory was among the first to demonstrate that multiple forms of regulated necrosis contribute to retinal neuroinflammation and degeneration. Over the past 16 years, his work has been continuously funded by the National Eye Institute through multiple R01 awards. Most recently, his laboratory has pioneered investigations into epigenetic regulation of retinal degeneration development, supported by an NIH R01 grant focused on how DNA methylation and TET-dependent DNA demethylation control photoreceptor development. Today, he will present evidence that impaired DNA demethylation by genetic ablation of TET enzyme in retinal progenitor cells disrupts activation of key developmental genes, leading to abnormal photoreceptor maturation, functional blindness, and retinal dystrophy. Please join me in welcoming Dr. Dmitry Ivanov for his talk, Impaired DNA demethylation in retinal progenitor cells prevent photoreceptor development and leads to retinal dystrophy.
Thank you so much for this introduction, and actually it's a great honor to be here. And today I would like to tell you about a new, but I think it's very important, epigenetic mechanism responsible for the development and function of photoreceptors. Okay, in this presentation, I will show you that the development and function of photoreceptors depend entirely on a single epigenetic pathway, that is dependent DNA demethylation pathway. This pathway decides whether the photoreceptor will be a rod or a cone. This that dependent DNA demethylation pathway decides whether outer segments, connective cilium, and synaptic terminals will be formed and will be functional in photoreceptors. Same pathway decides whether photoreceptor will convert light into electrical signals via phototransduction and regenerate the light-sensitive pigments in the visual cycle. And since this existence of photoreceptors depend entirely on the TET-dependent DNA demethylation pathway, it's likely to be involved in inherited retinal disease. So, how can one pathway control everything? And my answer is epigenetics. So, what is epigenetics? As you know, upstream of any gene, we have a regulatory region known as a promoter, to which specific transcription factors bind to initiate gene expression. However, DNA does not exist in pure form in our cells. The nuclei of our cells contain chromatin, which is a complex of DNA and proteins, mostly histones, as shown here. Histones in DNA can be modified in chromatin, leading to emergence of epigenetic level of regulation of gene expression. So, initiation of gene expression depends on chromatin structure, which in turn depend on histone and DNA modifications. For example, if histone modifications lead to chromatin compaction, and as a result, gene promoter is unavailable for transcription factors to bind to, as it's shown here, then transcription will not occur. The necessary transcription factors are present, but expression does not. I think another example is important for this presentation. As you know, DNA consists of 4 nucleotides, one of which, cytosine, can be methylated. Methylation of cytosines can lead to chromatin compaction or can directly interfere with binding of transcription factors to DNA. This can lead in both cases to significant decrease or even absence of gene expression. So a high level of promoter methylation should lead to significant decrease or even absence of gene expression. Our objective was to understand whether DNA methylation influences photoreceptor development and function. As you know, early in development, the entire retina consists of retinal progenitor cells. In mice, for example, it's embryonic day 11. When retinal progenitor cells differentiate into all types of retinal neurons, including rod and cone photoreceptors, plus they differentiate also in Muller glia. So the best way to assess the impact of DNA methylation on photoreceptor development and function is to compare the DNA methylation patterns of retinal progenitor cells and photoreceptors. To this end, we investigated DNA methylation in mouse embryonic retina as a source of retinal progenitor cells. We also investigated DNA methylation in human embryonic retina as well as a source of retinal progenitor cells, but in this case, we used published data, next generation sequencing data. We also investigated DNA methylation in mature mouse retina as a source of photoreceptor cells because of rods account for about 70% of all retinal cells. We also investigated DNA methylation in isolated cone and rod photoreceptors. In this case, we also used published next generation sequencing data. Whole genome bisulfite sequencing was used to map DNA methylation across the entire genome at single base resolution. To analyze the whole genome bisulfite sequencing data, we used Bismarck, bisulfite read mapper, and methylation caller. We used MethylKit bioconductor package to study DNA methylation patterns, and we used DMR-seq bioconductor package to identify differentially methylated regions, or DMRs. Since I will be frequently using similar plots as shown here, I would like to explain in advance what they represent. First, on the top, the position of this. Okay, first, we have this blue lines and red lines. Blue lines, we're talking about level of methylation of this particular region in retinal progenitor cells, because of, in this case, we used mouse embryonic retina as a source of retinal progenitor cells. And red lines, they indicate a level of methylation in rod photoreceptors. This is a level of methylation in DNA in this region. The x-axis represents the position of its cytosines on the genome, and y-axis represents level of methylation. It means it's 100, 75%, 50%, and 25%, etc. Also, you can see the position of this DMR in the genome and its statistical significance are indicated at the top. This is position and this is FDR. What it means, how exactly the interpretation of these plots? It's quite simple. It means this gene, in this case rhodopsin, is highly, and promoter of this gene, rhodopsin, highly methylated in retinal progenitor cells, and methylation of this gene, rhodopsin, very low in rod photoreceptors. Same story here. In this case, only in blue lines related to methylation of DNA in cone photoreceptors. You can see methylation of DNA in cone photoreceptor in this region very high, and methylation of rhodopsin gene very low in rod photoreceptors. Okay, by studying DNA methylation in embryonic and mature retinas, we discovered that promoters not just one or two, but many genes critical for photoreceptor development and function were highly methylated in retinal progenitor cells. Methylation, and in this case, I'm talking about not only mice, but also in humans. Methylation of the promoters of the same genes was low in rod photoreceptors. So these genes include NR2E3 and NRL, which are required for rod specification. These genes also include genes required for outer segment formation and function, as it's shown here, for visual cycle, for connecting cilium formation and function, for phototransduction. Actually, it's a lot of genes required for phototransduction. Also, genes required for synapse formation and function. And again, I would like to tell you all these genes, promoters, all of these genes, highly metilated in retinal progenitor cells. And methylation, all these genes very low in rod photoreceptors. So from this data alone, you can see how the single epigenetic pathway can control the entire photoreceptor phenotype. Okay. Next, by comparing DNA methylation patterns in retinal progenitor cells, in rods and rod and con photoreceptors isolated from mature retina, we identified even more highly methylated in retinal progenitor cells genes. It was 43. We found that 14 of these genes are demethylated only during rod differentiation. 3 of these genes are demethylated only during con differentiation. And 26 of these genes are demethylated during differentiation of both rod and con photoreceptors. And I would like to show you some examples. In this case, for example, NR2E3, which required for rod specification. This gene, in promoter of this gene, is highly methylated in retinal progenitor cells and highly methylated in con photoreceptors. And methylation of this gene very low in rod photoreceptors. Next example we already discussed. This is rhodopsin. And another example, for example, PDSXB. PDSXB, it's a gene required for rod phototransduction, highly methylated in progenitors, highly methylated in con photoreceptors, and methylation of this gene very low in rod photoreceptors. In case of genes which demethylated only in cons, as example, it's PDSXC, which required for con phototransduction. And how you can see methylation of this gene very high in progenitors and very high in rod photoreceptors, but very low in con photoreceptors. In case of genes which demethylated during differentiation both rods and cons, I think it's a very good example PRPH2, which required for outer segment formation and function. In other name, it's RDS, unofficial name. And you can see this gene highly methylated in retinal progenitor cells, and methylation of this gene very low in this case rod photoreceptors, and in this case con photoreceptors. Recoverin required for phototransduction, highly methylated in progenitors, and methylation very low in rod and con photoreceptors. Another gene, RBP3, required for visual cycle, very important gene in case of visual cycle. And methylation of this gene very high in progenitors and very low in rod and con photoreceptors. So DNA demethylation, it looks like, occurs differently during retinal progenitor cell differentiation into cons and into rods. In cons, many genes specific only to rod photoreceptors are not demethylated. It means all these genes highly methylated in con photoreceptors and mature con photoreceptors. I think it's also very important to note that the promoters of many genes involved in inherited retinal disease are highly methylated in retinal progenitor cells and demethylated in mature retina. And actually, it's not entirely surprising, since many of these genes are involved in development and function of photoreceptors. As example, retinitis pigmentosa, we're talking about genes like NRL and NR2E3, we already discussed it. Rhodopsin, PD6A, PD6B, PD6G, CNGA, etc., PRPH2. Also, I think it will be interesting to know that methylation of many, but not all, photoreceptor genes was significantly increased early in mouse embryonic development during the transition from the inner cell mass stage to the epiblast stage. And again, we have same genes, NR2E3, NRL, a lot of genes required for rod phototransduction, including rhodopsin, and a lot of genes required for con phototransduction. Okay. Highly methylated photoreceptor gene promoters are demethylated during photoreceptor development. What do we know about pathways responsible for DNA methylation and DNA demethylation? First, the DNA methyltransferase family is responsible for methylation of cytosines in DNA. When 1011 translocation or TET enzymes modify methylated cytosines to such an extent that DNA repair systems the DNA repair system recognized them as damaged nucleotides and replaced them with normal cytosines. And that family was discovered, in this case, it's TET1, TET2, TET3 genes. This TET family was discovered by Dr. Anjana Rao in 2009. Okay, so TET enzymes must be involved in the process of DNA demethylation during differentiation of retinal progenitor cells into photoreceptors. What happens if we inactivate TET enzymes in retinal progenitor cells? If a TET-dependent DNA demethylation pathway controls the activity of genes necessary for the formation of the functional outer segments and functional synaptic terminals, presynaptic terminals, in developing photoreceptors, when by inactivating TET enzymes or this pathway in retinal progenitor cells, we can expect the appearance of cells in the retina lacking outer segments and synaptic terminals, as it's shown here. This result will indicate that the TET-dependent DNA demethylation pathway exerts global control over photoreceptor development and function. Okay, this is just our expectation. What actually happens if we inactivate TET enzymes in retinal progenitor cells? In this regard, we need to create TET conditional knockout. And since in mammals all three TET genes are highly expressed in the developing retina, we created mice in which all three TET genes were inactivated in retinal progenitor cells. This is CH-extent mice. To generate these mice, we use triple floxed mice, and we also use CH-extent Cre mice, in which Cre recombinase is expressed in retinal progenitor cells. Okay. As you can see, our prediction was correct. First, this is outer plexiform layer, and this is outer plexiform layer. This is TET control animals, this is TET control animals, and CH-extent again control animals, and this is experimental animal. I just would like to remind that the outer plexiform layer is formed by synaptic connections between photoreceptors and bipolar and horizontal cells. And how you can see, outer plexiform layer present here in this control, and actually outer plexiform layer is absent in experimental retinas. By the way, I forgot to tell, it's 3 months old animals. This is 3 months old animals. Same story in case of the outer segments. The outer segments also absent in experimental retinas. This is beautiful outer segments, cone and rod outer segments, and nothing is here in control animals. We also used ERG tests to show that the deficient animals, control animals, CH-extent TET or TET deficient animals, were functionally blind. It means that the photoreceptors of these animals were non-functional. In this case, we analyzed 3 months old animals, 1 month old animals, always these animals were blind. Of course, how exactly these animals can see if they don't have outer segments and it look like presynaptic terminals or photoreceptor presynaptic terminals. Okay, since CH-extent Cre animals were previously very well investigated and did not differ much from TET mice, we decided to use only CH-extent TET as experimental animals and TET as a controls. So, as you can see, the thickness of the outer nuclear layer in experimental retinas is quite close to the thickness of the outer nuclear layer in control retinas in the first months of life of these animals. However, over time, the outer nuclear layer of TET deficient retinas becomes increasingly thinner, indicating the death of photoreceptors. At the same time, compared to the outer nuclear layer, significant pathological abnormalities in the structure of the outer plexiform layer, as you can see it here, and outer segments can be observed from the early stages of development of experimental animals. So, we used transmission electron microscopy to study the changes that occur in the outer plexiform layer and the outer segments of 1 month old animals. Okay, In case of presynaptic terminals of rods and con photoreceptors, actually they are easily distinguishable and quite simple to observe using electron microscopy in outer plexiform layer. This is example of con presynaptic terminal, and this is example of rod presynaptic terminal. And as you can see, many rod synapses or presynaptic terminals are present in the outer plexiform layer of a control retinas. You can see this is beautiful flowers, and by morphology, by definition, this is rod presynaptic terminals. At the same time, we were practically unable to detect any rod presynaptic terminals in the experimental retinas, but we were able to detect some cone presynaptic terminals, but they looked really abnormal. It means we think this is cone presynaptic terminals, but it's hard to say. Anyway. Next, we also studied the outer segments of the photoreceptors of control and experimental animals using transmission electron microscopy. So, electron microscopy examination revealed well-developed outer segments in control animals. You can see this is outer segments, this is outer nuclear layer, and this is RP. But the outer segments were absent. We see outer nuclear layer, RP, and nothing in between. Or were underdeveloped in photoreceptors of 1 month old animals. You also can see these underdeveloped outer segments here. But in 3 months old and older deficient mice, even underdeveloped outer segments of photoreceptors, deficient photoreceptors, cannot be detected. The next question we would like to answer is how that enzymes affect the development of different subtypes of photoreceptors. Of course, you know, we have rod photoreceptors, plus we have S-con photoreceptors, M-con photoreceptors in mice, plus we have L-con photoreceptors in humans. But in mice, we only have S and M-con. In this regard, we used rhodopsin as a marker of rod photoreceptors, emopsin as a marker M-cons, S-opsin as a marker S-cons. What we found? We found that rhodopsin and emopsin are virtually undetectable in the retinas of TET deficient mice, experimental mice. You can see rhodopsin here, you can see this emopsin in case of control animals, but nothing is here. 1 month and 3 months old mice. In case of S-opsin, S-opsin can be found in the underdeveloped outer segment, as is shown here. And also interesting, S-opsin can be found around the nuclei of 1 month old animals. But in case of 3 months old animals, we only can detect S-opsin around the nuclei, as it's shown here. And of course, we cannot detect any outer segments in 3 months old animals. So, in this regard, if we limit ourselves to only these 3 markers, when the experimental retinas, it looks like contain only S-cons. Okay, is it really we have only cons in these TET deficient mice or TET deficient retinas, or we still have some rod photoreceptors? In this regard, one way to distinguish between rods and cons in outer nuclear layer is to analyze the chromatin structure in the nuclei. Con nuclei have a conventional chromatin organization: central euchromatin and peripheral heterochromatin. Rod nuclei have inverted chromatin organization, as it's shown here. And for example, this is typical rod nuclei, as it's shown in this image or here. So, our transmission electron microscopy results indicate that nuclei with rod-specific inverted architecture actually are present in the retinas of experimental animals. You can see them here or here. But in general, the number of nuclei with rod-specific inverted architecture was significantly lower in the retinas of experimental animals compared to controls. So, in this regard, rod photoreceptors are present in experimental deficient retinas, but the number of rods is significantly lower in experimental retinas compared to controls. Okay, we also use single nucleus RNA-seq analysis to independently evaluate the composition of experimental and control retinas. And in this case, what we found that the number of cones in CH-extended retinas, it's shown here, significantly exceeded the number of cones in control retinas. It means number of cones in experimental retinas significantly more. We have more cone photoreceptors in experimental retinas compared to controls. Rods are present in TET-deficient retinas, but the number of rods is significantly lower in experimental retinas compared to controls. So these results are consistent with that that presented in previous slides. It's also interesting, in experimental retinas, we also found cell clusters expressing just some rod or cone photoreceptor markers, but actually it's very low levels. We called these clusters rod-like and cone-like. And actually, this rod-like and cone-like cluster actually largest clusters in experimental retina. And you can see here, this is 0, this is a rod-like cluster. And number 4, this is cone-like cluster. They have a lot of cells in these clusters. We actually think that these clusters correspond to cells that start but failed to differentiate into photoreceptors, probably because of high methylation of the necessary genes. We also found a significant reduction in the number of all type of bipolar cells, as well as a decreased number of retinal ganglion cells and horizontal cells, plus some subtypes of amacrine cells, and plus annular glia. To independently confirm these observations, we performed additional tests. First of all, you can see it's interesting. The retina is smaller in experimental animals compared to controls in size. The number of retinal ganglion cells and the thickness of the optic nerves are also lower in experimental retinas compared to controls. The density, we also found that the density of TET-deficient CHAD-positive amacrine cells, we used CHAD as a marker of amacrine cells, but in this case it's marker of starburst amacrine cells. And the density of these cells was lower in ganglion cell layer, but interesting was higher in internuclear layer. And actually, it's if you look when we analyzed starburst amacrine cells, actually it looks like we have no big difference. Okay, anyway, I think what's interesting in this regard, the density of bipolar cells in the internuclear layer was lower in TET-deficient retinas compared to control, and bipolar cells were disorganized in the internuclear layer of TET-deficient retinas. The number of horizontal cells is low in experimental animals compared to controls, but this is, in our case, just more qualitative rather than quantitative result, because it was very hard to count horizontal cells in TET-deficient animals. But I think one of the important observation is related to the lamination of the inner plexiform layer. Dendritic processes of CHAD-positive cells were disorganized in the inner plexiform layer of 1-month-old animals. Okay, the inner plexiform layer of retina consists of 5 stratified subbands that segregate on and off visual pathways. And 3 types of retinal cells, retinal ganglion cells, bipolar cells, and amacrine cells, are responsible for elimination of the inner plexiform layer. And as you can see, the elimination of the inner plexiform layer was severely disrupted in TET-deficient animals. This result in itself, I think, is very important, since any defect in the elimination of the inner plexiform layer should lead to blindness. So TET-dependent DNA demethylation pathway is important for synaptic activity not only in the outer plexiform layer, but also in the inner plexiform layer. But we still have no information. We don't know which type of retinal cells are associated with this abnormality in the experimental retina. Our data also indicate that the dependent DNA demethylation pathway influences the development of Muller glia, as it's shown here. But anyway, taking all these results together, it looks like the TET-dependent DNA demethylation pathway contributes to the development of all retinal cell types. But our results also indicate that this pathway is the most significant contributor to rod and cone photoreceptor development and function. As you remember, TET-deficient photoreceptors lack synaptic terminals and also lack outer segments, without which these neurons are deprived of any function. Okay. To understand the molecular mechanisms leading to impaired photoreceptor development, we ask these questions. Does inactivation of TET enzymes in retinal progenitor cells result in the preservation of high levels of methylation of photoreceptor genes in TET-deficient retinas? And how exactly does this affect gene expression? So, to answer these questions, we, in this regard, compared DNA methylation patterns in experimental TET-deficient retinas, CHX10 TET, and control retinas. We used embryonic retinas as a source of retinal progenitor cells. And first, what we found, how we can see NRL and NR2E3 genes, they are highly methylated in retinal progenitor cells, but they are also highly methylated in TET-deficient retinas. And of course, methylation of these genes were very low in control retinas. And I just would like to remind you that NRL and NR2E3 transcription factors were required for rod specification. And in the absence of these transcription factors, photoreceptor precursors differentiate into cones. And again, I think what's interesting, you can if you compare pattern of methylation in case of retinal progenitor cells and TET-deficient retinas, it looks like they're quite similar. It means it looks like we don't have any demethylation at all when we activate TET enzymes in retinal progenitor cells. We also found that a significant number of genes important for rod and cone maturation and function remain highly methylated in TET-deficient retinas. These genes are responsible for formation of outer segments, for formation and function of outer segments, visual cycle, phototransduction again, connecting cilium formation and function, also synapse formation and function. And as example, I would like to show rhodopsin, highly methylated in progenitors and highly methylated in TET-deficient retinas. And of course, methylation very low in control retinas. PRPH2 gene required for outer segment formation function, highly methylated in progenitors and highly methylated in TET-deficient retinas. Recoverin, highly methylated again in progenitors and TET-deficient retinas, and methylation of this gene, which required for phototransduction, very low in this case, TET control retinas. Okay, next question, how exactly this high level of methylation can affect gene expression of all of these genes? In this regard, we used RNA-seq analysis, and we found that the expression of at least 70 genes critical for rod specification and rod and cone maturation and function, was significantly reduced in experimental TET-deficient retinas compared to controls. These genes are important for rod specification. It means, you remember I tell you about NRL and NR2E3, they highly methylated in TET-deficient retinas, and as a result, we have reduced expression of these genes. Plus, we also have reduced expression other transcription factors required for rod specification, like SRB and SAM7. We found reduced expression of genes required for visual cycle, phototransduction, outer segment formation and function, connecting cilium formation function, synapse formation function. And these are some examples. But what's interesting, I think you remember, I tell about 43 genes have to be, 43 genes are highly methylated in retinal progenitor cells, and must be demethylated to be activated in mature photoreceptors. But the expression of 70 genes, how you can see, was reduced in experimental retina compared to control. So we think that the TET-dependent DNA demethylation pathway regulates the gene expression directly and indirectly. And I think reduced expression of these transcription factors, NRL, NR2E3, and others, can explain how exactly TET-dependent DNA demethylation pathway indirectly controls photoreceptor gene expression. And also, I think, since expression of these transcription factors is important for rod specification, I think the reduced expression of these transcription factors, NRL, NR2E3, and others, can explain the reduced numbers of rod photoreceptors in experimental retinas. Also, okay, we have reduced level of expression of genes. What about proteins? What about protein level? In this regard, we use Western blot analysis, and how you can see NR2E3, PD6A, PD6B, PRPH2, NR2E3, it's rod specification, PD6B, 6A, and rhodopsin rod for transduction. PRPH2, it's outer segment formation. And how you can see, the level of these proteins are very low in experimental retinas compared to controls. I think these molecular changes can perfectly explain the impaired development and function of TET-deficient photoreceptors. I think also what important, expression of 56 genes, mutation in each of which lead to a form of inherited disease was significantly reduced. I think it's quite interesting result too. So how can DNA methylation affect the expression of photoreceptor genes? As you can see from these examples, DNA methylation is primarily reduced near the transcription start sites of these genes. For example, in case of NR2E3, this is transcription start site. This is this is control retinas. This is knockouts. And you see, in case of control retinas, we have significantly reduced level of methylation close to transcription start sites. NR2E3, PD6A, rhodopsin, PD6B, etc. So we think cytosine methylation in transcription start sites may interfere with efficient initiation of transcription of photoreceptor genes. But right now, it's just only hypothesis. Right now, it's just only idea. Okay. Our results suggest a mechanism by which the TET-dependent DNA demethylation pathway exert global control over photoreceptor development and function. We think that early in development, only genes required for cone maturation are demethylated and activated. Later in development, genes required for rod specification, as the NRL, NR2E3, are demethylated and activated, followed by genes required for rod maturation. Again, in this case, when I'm talking about rod maturation, I'm talking about outer segment formation, for transduction, etc. Disruption of this process results in cells lacking any photoreceptor function, as shown here. Okay, what actually we don't know? Actually, a lot. And this is just the beginning. We don't know how methylation of photoreceptor gene promoters prevent their activity. We don't know whether the demethylation of photoreceptor genes begins in retinal progenitor cells or occurs in photoreceptor precursors. We don't know whether the demethylation of photoreceptor genes occurs in one step or in several steps. And regarding this model, it's quite probably we're talking about several steps. Except for the fact that the TET enzymes are involved, we know nothing about other players involved in the demethylation of photoreceptor gene promoters. But I think transcription factors must be involved, and I need to explain this. How you can see, actually, TET enzymes, TET genes, they are housekeeping genes. It means you can find them everywhere, in any cells, in any cell. That enzymes, actually, what interesting, cannot bind to methylated DNA. It means these enzymes are responsible for DNA demethylation, but they cannot bind to methylated DNA. And actually, if you look these results, this information, it looks like that enzymes cannot bind to DNA at all. So, that enzymes require assistance from transcription factors that bind to that enzymes and deliver them to specific gene promoters for demethylation, as it's shown here. And the transcription factors are responsible for specificity of TET-dependent gene demethylation. As you can see in this picture, due to the specificity of this transcription factor, this green guy, the rhodopsin promoter is demethylated and activated, but PD6C promoter not. Another, I think, yeah. In this case, many groups of still unknown transcription factors orchestrate demethylation of highly methylated photoreceptor genes during con and rod photoreceptor development. For example, one group of such transcription factors is responsible for demethylation of genes required for rod specification. Another group of such transcription factors responsible for demethylation of genes required for outer segment formation function. Another group for transduction, etc. It means at least should exist at least six group of transcription factors. How many transcription factors in each group? I have no idea. Maybe one, maybe two, maybe three, maybe more. But we must identify these transcription factors if you want to understand how the dependent DNA demethylation pathway exert global control over photoreceptor development and function. And the question is not whether these transcription factors exist. The question is when we will find them. And actually, how you can see, it's like umbrella. This TET-dependent DNA demethylation pathway is like umbrella, which can cover many signaling cascades responsible for photoreceptor specification, outer segment formation and function, etc. And these particular transcription factors, they are responsible for specificity of this TET-dependent DNA demethylation pathway. Another reason why this is so important to identify these transcription factors is they are most likely involved in inherited retinal disease. For example, if mutations in this transcription factor responsible for demethylation of rhodopsin promoter, if we have mutation in this transcription factor, then this should lead to lack of rhodopsin expression, and as a result, to inherited retinal disease. It means, but what's interesting in this case, we're talking about inherited disease. Most likely, it will be retinitis pigmentosa. But in this case, this IRD is caused not by mutations in rhodopsin, but by high levels of methylation of promoter of this gene. And we call this form of IRD the epigenetic form of IRD. So we can distinguish two forms of IRD, genetic and epigenetic. Mutations which disrupt the activity of IRD gene, like rhodopsin, PD6A, PD6B, PRPH2, NRL, NR2F3, etc. So mutations that disrupt the activity of IRD gene lead to genomic form of IRD, genetic form of IRD. Mutations which disrupt the activity of transcription factor responsible for this IRD gene demethylation lead to epigenetic form of IRD. Genetic and epigenetic IRD are likely to be phenotypically similar. And the reason this form of IRD should be phenotypically similar is that the IRD gene is inactive in both cases. But the difference is that in the genetic form of IRD, the IRD gene is inactive due to mutation. But in the genetic form, epigenetic form of IRD, the IRD gene is inactive due to high level of methylation of its promoter. But again, we only can can find this epigenetic form of IRD when we can identify all these transcription factors, or at least some of these transcription factors. Okay. As a conclusion, many genes essential for photoreceptor specification and maturation function are highly methylated in the retinal progenitor cells and must be demethylated to be activated in mature photoreceptors. The TET-dependent DNA demethylation pathway is a master regulator of photoreceptor development and function. We must identify the transcription factors if you want to understand how the TET-dependent DNA demethylation pathway exerts global control over photoreceptor development and function. And actually, it's the reason why I quote Tolkien's The Lord of the Rings here. One ring to rule them all, one ring to find them, one ring to bring them all, and in the darkness bind them. In this case, darkness probably high level of methylation. Anyway, in this case, one pathway, one ring, one pathway, TET-dependent DNA demethylation pathway, can control many pathways or signaling cascades responsible for the receptor development and function. And also, I think it's very important to it looks like we have to distinguish two types of IRD, genetic and epigenetic. But it's a different story. Okay, but before concluding this presentation, I would like to acknowledge the laboratories that have contributed to this field. It looks like that the first mentions of the demethylation of some photoreceptor genes during retinal development can be found in these two publications. It's Dr. Anand Swarup lab publication in Cell Reports and Dr. Michael Dyer publication in Neuron. In case of Dr. Anand Swarup, they isolated rod photoreceptor precursors from postnatal day to retinas. And they found that methylation in these rod photoreceptor precursors of these two genes, rhodopsin and PD6P, still very high. And of course, the methylation of these promoters of this gene was low in mature rod photoreceptors. In case of Dr. Dyer lab, it looked at least one gene, PD6B, was highly methylated in mouse and human retinal progenitor cells. Also, Dr. Jeffrey Gross laboratory demonstrated the role of TET enzymes in the development of zebrafish retinas, and including in development of photoreceptors. His publication first is he published, for example, PLOS Genetics 2017. In our case, our lab discovered that not one, two, but dozens of genes must be demethylated during photoreceptor development in mammals, and TET enzymes are responsible for this process. And I think we are pleased that our observations have been independently confirmed by Dr. Clark's laboratory. They published it in PLOS Biology in 2025. We published in PNAS in March 2025, and we published it same year in PLOS Biology. Okay, I'm very grateful to everyone involved in this study, and I would like to express my special gratitude to Galina Dvoryanchikova, without whose efforts this study would not have been possible. And this study was supported by the NIH and by Research to Prevent Blindness. And thank you. I think I'm ready to answer your questions.
Dmitry, very clearly, this is a very important mechanism, how the retina is made. I have maybe I will start with two comments or questions, I should say, rather than comments. The one is, did you look in RPE? Did you look whether the same mechanism applies to RPE and enzyme involved in visual cycle?
Actually, we have this data, not exactly, because we just analyzed entire retina, the deficient retinas. In this case, we need to collect RP layer and analyze. We can do it, but we didn't. It means actually we can do it. It will be. You're absolutely right. It's very interesting. Yeah, absolutely. But we didn't.
That may be. And let's go not developmental, because there's not much we can do about that. You know, this is program formation of the retina. But did you look on more mature retina and to look on circadian rhythmicity, whether there is a changes in methylation during the day in the critical genes of photoreceptors as well as RPE?
No, we didn't. But I think because in this case, we only analyzed, like, we found a high level of methylation of these genes in retinal progenitor cells. And in case of mature photoreceptor and mature retina, it's always looked like methylation very low. Yes, it's if we analyze different time points, like day, night, but no, we never did it. Actually, probably it's quite interesting.
Yeah, Franz Winberg, UCI.
Mhm.
So you have these several TET genes. Is there like high redundancy between them, or do you think there are like different TET genes specifically demethylating certain genes?
I think it's quite possible because it was shown, for example, TET2 more required, like more important for demethylation, like enhancers. But anyway, yes, by definition we have redundancy, and it's reason why we need to inactivate all of these three genes. But actually, it looks like two genes, TET2 and TET3, more important, because if we inactivate two genes, TET2 and TET3, we already have significant changes. We already have significant damage and in case of outer segment, outer plexiform layer. It means probably TET2 and TET3 more important. But anyway, when we inactivate all of them, we have significant damage in retina in this regard.
Yeah, yeah.
But you're absolutely right. By definition, we have redundancy here. And probably TET1, TET2, TET3, they play maybe some different role. Maybe TET3 more important for demethylation promoters, TET1 may be more important for demethylation enhancers. But in our case, it was very hard to analyze what about enhancers in case of these genes, because we don't have so much information about this enhancers of these photoreceptor genes.
And other thing is conservation. So I guess between mouse and mammalians and all the vertebrates very concerned. But what about invertebrates and the photoreceptor development in those species?
I have no information. I think it will be interesting, but it's hard to say. I have no information.
Hey, Dr. Ivanov, Vladimir Kefolov, also in the audience. Thank you for your great talk. Following on Chris's question on the kind of the regulation of the methylation in the adults, have you looked, or anyone else, in what happens during retinal degeneration? And kind of related to this is, are the TETs expressed in the adults, and could demethylation also play a role in retinal degeneration?
I think these enzymes probably involved in aging process. One possibility, because it looks like with aging, its activity of these TET enzymes is going down, and a level of methylation just increased in promoters of genes. But another one, I think it was in case of glaucoma, for example, it was shown if we inactivated TET2, TET3. Okay, optic nerve regeneration. It looked like TET2 and probably TET3 required for optic nerve regeneration. And it was shown, I think it's Dr. Sinclair, I think, publication, when they activated TET2 and TET3 level in retinal ganglion cells, they found improvement in vision, etc. It looked like, yes, by definition, TET enzymes probably involved and important, and they help to survive cells. It's hard to say about retinal. It's hard to say about photoreceptors when we're talking about late stages, for example, age-related macular degeneration, etc. But yes, it looks like very important. But we need to learn more about, because again, this family was discovered just in 2009. We still need to learn a lot about this family, and in my opinion, it's quite amazing, quite interesting field to understand an epigenetics in particular, in how it's role of these TET enzymes in case of development, and plus in case of degeneration, in retinal degeneration, photoreceptor degeneration in this regard.
Okay, thank you. And my follow-up question is about this really interesting idea about the epigenetic form of inherited retinal diseases, degeneration. So are there already known diseases linked to the epigenetic modulation of demethylation? And generally, what do you have a sense of what is the relative contribution of mutations related to promoters and this epigenetic idea versus kind of the typical inherited retinal diseases that we think about?
I think we still have no idea about. We still need to. It's just right now. It's just idea. It looks like because before we can identify transcription factors which help that enzymes to demethylate promoters of these photoreceptor genes, I think we cannot say these forms of inherited epigenetic form of inherited disease exist. It means first we need to find these transcription factors, and when we already, if we can find mutations in these transcription factors, we can say yes, this epigenetic form of exist. And something like this. I hope I answer your question.
Okay, yes, thank you. Yeah.
Hi, my name is Kim. I'm from Dr. Pacheski lab, and your talk is extremely exciting and interesting. And my question is, in the model that you have with TET deficient, have you ever tried to rescue that by overexpress one TET or all three TET?
No, we never did it. But the problem here, because we have to understand, when it's impossible to rescue, because okay, in this regard, in this case, TET enzymes, we inactivated completely TET enzymes, and actually, in case of demethylation, we need specific transcription factors, and it looks like these transcription factors, which are responsible for DNA demethylation, it's quite possible these transcription factors have high expression in particular time point. It means in this particular time window, we have expression of these transcription factors, and they're responsible for demethylation of this particular gene, for example, rhodopsin. But later, maybe this transcription factor already have expression, low expression, no expression at all. It means it's impossible to save these photoreceptors just, for example, to increase expression of TET1, TET2, or TET3, because we need these specific transcription factors. And if these transcription factors they have high expression before, it means nothing can help. But I agree with you. Actually, it's possible if we can, for example, to try to do it, because maybe these transcription factors still exist in adult photoreceptor, mature photoreceptor. Yes, probably it can help. But my expectation, I think, all these transcription factors which we discussed, they probably activated at some particular time point. And when they just expression going down, when some another group of transcription factor activated, and as a result, they can regulate this, for example, con differentiation photoreceptor plus rod differentiation photoreceptors. It means you're absolutely right. It means it's a good idea to try it to increase expression of these genes, but I think my expectation probably we cannot see anything. It means because it's very important. Again, I repeated many times: transcription factors. We need to identify first these transcription factors. We need to understand expression of these transcription factors in development, rod and con development. And after this, we already have idea: can we save these photoreceptors in mature retinas, or we cannot do it? I hope I answered your question.
Yes, you did. My following question that in the case when age-related degeneration, where that DNA methylation also being dysregulated, so would you think that if we express one of these TET in there, now the transcription factor of some of them is no longer available because past certain time point, would there be any hope that if you re-express TET protein that will rescue some level, or?
Yes, probably not. But in this case, I think right now exists CRISP technology. Actually, right now exist systems which you can use CRISP technology, plus you can put TET1 protein. It means, for example, if you need to demethylate particular gene, it's highly methylate. Maybe in this case, better to use this CRISP technology compared to just overexpress with TET enzymes, TET genes. Maybe it will be best solution.
Thank you.
Oh, thank you.
Hi, this is Rafał from Krzysztof Palczewski lab. In the topic of transcription factors, it's interesting to me that there is this heterochromatin that should be poorly accessible, and I have a question: what would be the starting point? Is there like a specific property of the transcription factors, or what is the mechanism to actually start the transition from heterochromatin to euchromatin? Thank you.
It's hard to say. It's hard to answer to me. It's not simple question. It's like basic question. How what has happened? I think it's to me hard to answer this question in this regard. Yeah. Right now exist many different mechanisms which responsible for like transition between chromatin and euchromatin. But in this particular case, I think it will be hard to answer this question.
Thank you.
So hi, this is Tripti from Dr. Audron Lapinate's lab. So thank you for the great talk. I have a question. Like, since you say that the retinal progenitors are not maturing and they are not able to form rods, did you see the expression of progenitors like OTX2 or CRX by immunohistochemistry in the TET knockout in the TET animals? Like, was there an increase in their expression, which we normally expect?
You talking about which gene?
OTX2, CRX.
Okay. I understand. Actually, OTX2, we have normal expression, and CRX, we have normal expression. It's no changes in expression at all. And by the way, keep in mind, OTX2 actually, and by the way, what important, in case of OTX2, methylation of this promoter of OTX2 very low in retinal progenitor cells.
Okay.
It means it's no problem for photoreceptor to start differentiation. It means no problem for rod and con photoreceptor because OTX2 required for this. But to finish this differentiation, you need other genes required, for example, for con maturation, and you need genes required for rod specification, like NRL and R2-3, and rod maturation. And in this case, because of we have very high level of methylation of these genes, it looked like photoreceptor just start differentiation, but they cannot finish it. And probably this is the reason why at some moment they start to just die. This is underdeveloped photoreceptors which doesn't have any synaptic terminals. They don't have outer segments, and it's something. It's not exactly even photoreceptor, because how exactly you can be photoreceptor if you don't have synaptic terminals and outer segments? It means everything. OTX2 probably push differentiation of retinal progenitor cells in direction of photoreceptors, but they cannot finish this differentiation because of a high level of methylation of these genes.
Okay, so following to this, like since we know that cone forms first and then rods, so because of which, like since NRL and NR2E3 expression is not seen and it is not expressed maybe, so based on which can you consider that cone differentiation was more because they are not able to form rods now, and therefore you see more cones and not rods?
You're absolutely right. Yes, probably it's quite possible because these cells cannot differentiate into rods. Yeah, it's the reason why we have some more cones. Yeah, you're absolutely right. And by the way, S cones, because we don't have M cones. It means it's quite. By the way, remember, S cones is default state.
First,
it means, yeah. You're absolutely
you.
right. Yeah.
Thank you.
Hello. I have a question about M-cons. So they appear to have a different transcription network factors. So what happens to them in your knockout animals?
We actually, I have no idea, because in general, what we analyzed, it's like genes required for both cons and S cons and M cons. And for example, gene like I think THRB, thyroid hormone. Anyway, it means we only analyze just like rod photoreceptors markers and con photoreceptor markers, and we never separated between S and M con. Only in this case, M-absine, we have very significantly reduced gene expression in case of M-absine. Plus, what's interesting, we have significantly reduced expression of some genes required for con maturation, like PD6H, which required for con specification. But again, it's hard to say. To me, it's hard to separate between S and con. I think it's very interesting question, and we need to check with other genes, but we didn't. It's very interesting question. Thank you.
Dmitry, really, this is very important mechanism, and we're very delighted to have you here to tell us about your work. And we will finish with the final question from Dr. Chen.
Hey, Dmitry, it's really nice talk. So I'm thinking about, the one of the phenotype you show briefly is the overall the size or the number of cells, right? In the retina is smaller in the knockout. And then so is that because the progenitor cell proliferation, the pool becomes smaller overall? And any insight on why is that phenotype?
Actually, it's a beautiful question, and it's really interesting what is going on with these retinal progenitor cells. And we still need to learn this, because right now it's hard to say. It's quite possible it's because of retinal progenitor cells, maybe rate proliferation of these cells they proliferate like slower or something. It's quite possible. We need to learn more about this. It's quite interesting. That's absolutely important question. And we right now we're just working. We try to figure out what is going on here. But this question, because to me, I really was surprised. We, in this regard, retina was really small in these animals compared to controls. And how exactly we can explain this small size? Plus, number of many cells also was very low. It means it's quite interesting. And I think retinal progenitor cells, proliferation of this rate of proliferation, I think it's a very important question. And we need to answer this question.
All right. Thank you so much. Please join me to thank our speaker today.
Thank you.